Dear colleagues, welcome to today's webinar. Today we start with the second part of the PEPANCO. Last year, in the first part we discussed general regulatory principles. Now we want to focus on oncology.
But before we get into the actual content, let us take care of the formalities. This presentation is the sole responsibility of the authors. We are not speaking on behalf of the EMA, or our national regulatory authorities, or the CHMP. We are all employees of a regulatory authority and have no conflicts of interest.
Today we want to offer you a high-level overview of the medicines authorized by the European Medicines Agency for the treatment of cancer. In the introduction, I will tell how we have structured this overview according to the pharmacological mechanism, the mode of action of the drugs. In other words, according to drug classes. We will therefore discuss active substances with the same principle of action together, even if their clinical use may be completely different. Based on their molecular targets and pharmacological mechanism, we can put all the medicines in four broad categories: drugs whose efficacy is mediated by the immune system, drugs that intervene in the control of growth through signaling, drugs that target the genetic material of the cell, and finally the colorful category others, with everything that we cannot categorize in any other way.
A brief look into history. Regulation 726/2004 stipulates that innovative medicinal products for the treatment of cancer can only be authorized using the EMA's centralization procedure, making the centralized procedure mandatory for anti-cancer medicines. The voluntary use of the centralized procedure was possible before, but until then, most medicines for the treatment of cancer were authorized nationally by the individual member states of the European Union. Before Regulation 726/2004 introduced the mandatory scope, as little as 14 active substances for the treatment of cancer had been authorized in the centralized European procedure. However, the four categories into which we divide the mechanisms can already be seen, and most of them can be found in the category genome, i.e. in the category for substances that act on the genome of the cancer cell.
Today, things look very different. More and more products with new mode of action are being introduced into clinical practice. Since 2014, the number of new molecular targets and MOA has constantly increased. EMA has authorized almost 200 active substances, with 75 different molecular targets or pharmacological mechanisms. All substances and classes are here on this busy slide, arranged chronologically. Pharmacological classes are used as building blocks forming columns or towers, starting at the bottom and adding new classes or substances on top. Of course, this is not meant to be read here, but is for your reference in the slide deck you have or will receive, together with a list of abbreviations for the class names. For today, we will focus on the groups, that is the columns or towers, and pick only a few examples for each category where we go more into the details. While the high number and heterogeneity of target shows how greatly the armamentarium for the pharmacological treatment of cancer has expanded over the last 2 decades, there have been apparent shifts in the preferred pharmacological mechanisms or molecular targets of anti-cancer medicines. In the last few years, decisive new developments took place primarily in the field of immunotherapy and cell signaling, whereas the last new class in the category genome was introduced 10 years ago. Cancer immunotherapy is currently the most interesting field. Within the category immunology, we have 4 groups: hematologic cell surface antigens, immunomodulators, checkpoint inhibitors, and antigens in solid tumors. While cancer immunotherapy is currently probably the most trendy category, it is remarkable how long the approach of immunological cancer therapy has been pursued. The best-known example is probably Coley's work at the end of the 19th century. Nevertheless, since we are at the BFARM in the city of Bonn, I would like to point out that it is assumed that Coley built on the work of Felizen and that Felizen pursued experimental approaches by Bush, and that Bush was a clinician and scientist at the University of Bonn, so, if you like, not the father, but the grandfather of the immunological therapy of cancer is from Bonn. And after this promotional digression, I hand over for a more scientific and detailed look at the category immunotherapy.
Thank you. In the category immunology, with the 4 groups, or columns or towers, 2 groups are particularly noteworthy here. Firstly, in the immunology category, we have the group of immune checkpoint inhibitors, with their very high clinical relevance and broad range of use against a variety of different tumors. Secondly, we have the tallest tower with the largest number of different molecular targets, the group of substances that target hematological cell surface antigens. Here you have an overview for the immunology category, with all active substances that have been authorized by the centralized procedure and are still on the market, with product name and year of authorization. Of the 4 groups, hematologic cell surface antigens, immunomodulators, checkpoint inhibitors, and antigens in solid tumors, the obviously largest group is hematologic cell surface antigens. Half of all molecular targets and more than half of all innovative new active substances or products fall into this group. The dynamic development of this group is explained by the special nature of hematological neoplasms. Hematological cell surface antigens lend themselves as pharmacological or immunological targets because it is not necessary to find tumor-specific targets. Cell type-specific targets will be sufficient. In the case of hematological neoplasms, it is possible to eliminate both the cancer cells and the associated normal, non-neoplastic cells, as long as the stem cells are preserved, or can be replaced by stem cell transfer if necessary. This is a major difference to other organs. For example, in the case of glioma, I cannot eliminate all glial cells. In the case of liver cancer, I cannot eliminate all liver cells. At variants, differently, I can eliminate all blood cells of a certain type as long as they can be regenerated from the stem cells. What is remarkable here is the high degree of particularization of neoplastic diseases of the blood system, i.e. the many different types of cancer based on the different stages of differentiation of the same cell line. Firstly, this leads to the high number of different pharmacological targets in this group. And secondly, because of this high level of particularization, we have almost only orphan drugs in this group. If we could simply group all lymphomas together into one disease entity, just as we can group all prostate cancers together, there would of course be fewer that could be considered an orphan disease. Let's have a closer look at the antibodies directed against CD20. This oldest class with the target CD20 is a good example of how therapy with mAbs has evolved over the years. The first product, MabThera, introduced a long time ago in 1998, was still a simple antibody. Zevalin was already a conjugated antibody to be used with radioactive yttrium. Gazivera was an improved, glycoengineered mAb. And then after an 8-year gap, we got Lunsumio and Columvi and Tepkinly, all 3 almost at the same time, and all 3 are bispecific T-cell engagers BiTE, working on a quite new level. By the way, we are currently seeing a boom in bispecific T-cell engagers, here in blue. Although Blincyto was already approved in 2015, others did not follow until 2022 and 2023, but then in rather impressive numbers. It should be noted that Kimmtrak is an exception. Kimmtrak is a fusion protein, not a true bispecific antibody. The group of hematologic cell surface antigens is also outstanding for other new developments, and it is the spearhead for new developments. ATMPs for the treatment of cancer, here in green, more precisely CAR T cells, are also found exclusively in this group. Although the first immunotherapy, CD20, was approved in 1998, it took more than 10 years before a sustainable development began, with more monoclonals bringing immunotherapies in clinical use. 2011 saw the first CPI, 2012 the first conjugated antibody against CD30, and 2015 the first antibody directed against an antigen on a solid tumor. Antibodies whose effect is based more on the interruption of cell signaling than on the induction of immunological reactions will be considered in the signaling category, although the pharmacological effects of these antibodies may have an immunological component, too.
In this more than 10-year gap between rituximab and the groundbreaking introduction of the first CPI, various immunomodulators were introduced and tried to fill the gap mostly with only moderate success. It is noteworthy that both Mepact and Baraman were developed for the treatment of sarcomas, precisely for the tumors that Coley was already working on more than 100 years ago. The most important of these is certainly the thalidomide class, and even though thalidomide has a very troubled history today as a treatment for multiple myeloma, it can also be seen as a prime example of repurposing. And here the product name Celgene is not entirely correct, as this medicine is sold as Celgene thalidomide. The most significant development in this field of cancer immunotherapy is certainly the introduction of the immune checkpoint inhibitors. The first immune checkpoint inhibitor was Yervoy, ipilimumab, back in 2011. However, checkpoint inhibitors only really gained momentum with the PD-1 antibodies in 2015. The fundamental difference to the hematological cell surface antigens is obvious. While these are each intended for the treatment of a very specific neoplastic disease, checkpoint inhibitors can usually be used against a broad variety of different tumors. Accordingly, we also see a very high level of post-authorization activity for extension of the indication for these drugs. Blincyto, this first-in-class anti-CD19 bispecific antibody, approved in 2015, had 38 type 2 variations. Keytruda and Opdivo 130, both approved in 2015, had each about 140 type 2 variations. Currently, the clinically by far most important classes are anti-PD-1, and to a lesser extent, anti-PD-L1 checkpoint inhibitors. The binding of PD-L1 to PD-1 keeps T cells from killing tumor cells in the body. Blocking the binding of PD-L1 to PD-1 with an immune checkpoint inhibitor, such as anti-PD-L1 or anti-PD-1 antibodies, allows the T cells to kill tumor cells. Biomarkers allow the selection of patients for whom the checkpoint inhibitors work best. Sometimes the percentage of tumor cells that stain positive, the tumor's proportion score, TPS, is used as a biomarker. In other indications, the percentage of tumor cells as well as immune cells that stain positive, the combined positive score, CPS, is used. For example, TPS is commonly used in lung cancer, while CPS is used in some other cancers such as gastric cancer. The binding of B7-1 or B7-2 to CTLA-4 keeps the T cells in the inactive state, so they are not able to kill tumor cells in the body. Blocking the binding of B7-1 or B7-2 to CTLA-4 with an immune checkpoint inhibitor, such as an anti-CTLA-4 antibody, allows the T cells to be active and to kill tumor cells. The same principle works for all checkpoint inhibitors. In this case, LAG3 is expressed on the cell membranes of activated T cells. It is also expressed on natural killer cells, B cells, and dendritic cells. The binding of B7-1 or B7-2 to CTLA-4 keeps the T cells in the inactive state, so they are not able to kill tumor cells in the body. And again, blocking the binding of B7-1 or B7-2 to CTLA-4 with an immune checkpoint inhibitor, such as an anti-CTLA-4 antibody, allows the T cells to be active and to kill tumor cells. While the most significant development in this field of cancer immunotherapy is certainly the checkpoint inhibitors, and while there has been a steady stream of new pharmacological targets in the group of hematologic cell surface antigens for over 10 years, antigens from solid tumors are comparatively rarely used as targets for immunotherapy. In contrast to the checkpoint inhibitors, we are dealing here with narrow niche indications, the best example being Kimmtrak, which can only be used for melanoma of the eye and in patients with a specific HLA type. On the other hand, Trodelvy, with the indication triple-negative breast cancer, is an encouraging example that this approach is also possible for solid tumors with a high prevalence. And for insiders, The difference between Qarziba and Unituxin is that the original dinutuximab in Unituxin was from a myeloma cell line, whereas the dinutuximab beta is from Chinese hamster ovary cells. And with this we leave the immunotherapy field and go to the next category.
Thank you. Next, we will look into the category signaling, with the groups endocrine, protein kinases, and the mixed bag we called others, where we put all signaling that is neither sex hormone nor protein kinase. Cell signaling is the category with the most products or active substances, with the largest number of different mechanisms of action or molecular pharmacological target structures. The principle of interrupting or modulating the reception or transmission of growth signals is comparatively old. The principle of blocking hormonal signals was introduced into cancer therapy at the end of the 19th century through oophorectomy for breast cancer. In the first half of the 20th century, the same principle of castration was introduced in men with prostate cancer.
Here you have an overview for the cell signaling category. With all active substances that have been authorized by the CP and are still on the market, with product name and year of authorization. The endocrine treatment of cancer can be regarded as a special and the most early form of targeting the cell signaling, even if introduced before the concept of cell signaling entered the mainstream. Endocrine treatments of cancer have been developed and used long before signaling overall became a frequent target. Many of them were already authorized by national regulatory authorities in the last century and therefore do not appear here. And there is nothing new, no new targets or mechanisms for the past 10 years. A very large group, or more precisely, two very large groups are the protein kinases. In our structured overview, we therefore differentiate, depending on the target location, for example intracellular or membrane-bound with an extracellular domain, two groups are defined. While intracellular protein kinases are inaccessible for antibodies, a high number of different anti-cancer medicines, including small molecules and monoclonal antibodies, can be used for the inhibition of cell membrane-bound protein kinases with an extracellular domain. Accordingly, macromolecules such as monoclonal antibodies, here in blue, conjugated monoclonal antibodies, here in green, or fusion proteins, here in pink, can be used only to target protein kinases on the cell membrane, not intracellular protein kinases. At least that is current status. However, this may not remain an unchangeable limitation for the use of monoclonals. Recent research indicate that IgA could also make it possible to reach intracellular targets with antibodies, and this might open the door to a most promising widening of the use of antibodies. By the way, here is an inconsistency in our systematic structure. The vascular endothelial growth factor, VEGF receptor, is of course a tyrosine kinase and is the pharmacological target for the whole range of substances as shown in the box on the right. With the exception of ramucirumab, they are all small molecule substances that often act not only on the VEGF receptor but also on other protein kinases. What is not quite correct, but pragmatic, is that we have also included substances here that target the ligand VEGF rather than the receptor. The rationale is that they target the same signaling chain. Nevertheless, admittedly, VEGF is not a protein kinase. With protein kinases, we can clearly show how progress and further development is possible not only with new molecular targets but as well within a pharmacological class, and that other, newer products in the same class need not be a me-too. Trastuzumab was the first substance with the HER2 as target. This was followed by lapatinib, not an antibody but a small molecule, before Kadcyla, the first conjugated antibody against HER2, was approved in 2013. Almost at the same time, pertuzumab was approved, which binds to a different HER2 site than trastuzumab, and can therefore be used in combination with trastuzumab. A similarly favorable development can also be seen with the successor products to Gleevec, imatinib. Resistance was overcome with Sprycel, dasatinib, and Tasigna, nilotinib. The different side effect profiles of the two substances, nilotinib with the problem of cardiac risk and dasatinib with the problem of fluid retention, allowed the selection of a therapy that is better tailored to the individual patient. Pontinib, iclusig then proved effective against the problem mutation T314I. The latest in the sequence, Scemblix, asciminib, which has now been approved with a significant time lag from the others, uses a different binding site, namely the myristoyl pocket, at variance with all other substances in this class, which target the ATP binding site. By the way, it is an amazing parallelity that both the groundbreaking small molecule imatinib and the groundbreaking antibody trastuzumab are followed several years, 21 years and 13 years, later by another, small molecule for imatinib, antibody for trastuzumab, that have a different binding site on the target molecule, target a different part of the molecule with a different binding site, which again shows that that same class does not mean me too, not even when we have two basic. That is not conjugated, antibodies that both target the same receptor. The immunotherapy of cancer had their game changer with the introduction of the checkpoint inhibitors. I dare to say that we have another game changer here with the acceptance of histology-independent, tissue-agnostic indications. Europe may be more hesitant here than the US, nevertheless, Vitrakvi, larotrectinib and roseltrek, entrectinib probably opened the door for the evolution even of new disease entities based no longer on histopathology, or on morphology, but on genotype, on genetic biomarkers. And this has consequences not only for new medicines, that may well change the use of previously approved products, too. Currently, Retsevmo is under assessment for adding an agnostic indication to its current classical, histology-based indications. Allow me a short digression into somatics. I hope you will forgive that I did not follow the mainstream when I used the expression histology or tissue agnostic. Apparently, tumor agnostic is the preferred term both in the literature in PubMed and in whatever is found by Google, and I will not elaborate why I think tumor agnostic is not really the best, as long as we all have the same understanding what is meant. Finally, other signaling. A rather heterogeneous group. Skiva, denosumab does not really directly target the tumor cells but the bone, strengthening the bone. This is why denosumab is used for the treatment of osteoporosis, too, and that way slowing the growth of bone metastasis. Substances that target the hedgehog pathway are a good example how substances from the very same class may have very different indications, with Durismo, Glastagib used for the treatment of acute myeloid leukemia at variance with Odomzo, Sonidegib and Ariveg, Vismodegib, which are used for the treatment of basal cell carcinoma. And as the latest in other signaling, we have the KRAS targeting Lamicras and Krazati. Admittedly, efficacy may be somewhat less than initially hoped for, but still interesting. When I worked at the German Cancer Research Center a very long time ago, well before the turn of the century, RAS was the prime example of undruggable. And now we have the second active ingredient that works.
Well, lesson learned. Don't take no for an answer, not even from nature. And with this profundity, we leave the cell signaling category and continue with the two smaller categories, substances targeting, directly or indirectly, the DNA of the cancer cells. Before we go to the mixed bag of others, where we put everything that we could not place in any of the other categories. Let's start with the category genome, with the three groups. Mitotic apparatus, DNA integrity, DNA synthesis, and antimetabolites. The origins of many substances in this group go back to the Second World War, to chemical warfare agents. Gilman and Goodman developed the first modern chemotherapeutic agent from mustard gas in the 1940s. And even today, substances from this class are still part of the standard repertoire of chemotherapy, even if melphalan has recently been withdrawn in the USA. Many products in this category have gone through national authorization procedures. Many of these products were authorized in national procedures long before the European authorization was created. This is why the collection of classes that we have here is rather incomplete in contrast to the other categories. Nevertheless, here you have an overview for the genome category, with all active substances that have been authorized by EMA's decentralized procedure and are still on the market, with product name and year of authorization. These active substances that simply have DNA as a target and that often damage all proliferating cells, we cannot consider a targeted therapy.
With one exception. The last new class was the poly-ADP ribose polymerase inhibitors in 2014. This class is the only one in the genome category that can be considered a targeted therapy. The indication, at least for some tumor types, requires the detection of a BRCA mutation. Strangely, however, with the newer products of this class, the indications seem to become not more precise, but less targeted. For both breast cancer and prostate cancer, the first-in-class product, Lynparza, olaparib, requires the detection of a corresponding mutation for therapy. However, the successor products for both tumor types have a broad indication that no longer requires this mutation test. Even with old therapeutic approaches, such as antimetabolites, there are still relevant new developments. For example, in the class of pyrimidine antimetabolites, fixed-dose combinations have been developed that enable oral therapy. Admittedly not an exciting new pharmacological mechanism, but nevertheless quite relevant for patients.
The last category, others, in which we have categorized everything that does not yet fit into one of the major patterns, appears quite heterogeneous. Heterogeneous indeed, but attractive. Perhaps we will even see the development of a new category here in the future, but at present it is, at best, provisional and speculative, a group of active substances that target housekeeping metabolism. Here you have an overview for this category, with all active substances that have been authorized by the centralized procedure and are still on the market, with product name and year of authorization. The wide range of substances in this group is remarkable. We have here modern, complex therapeutic approaches such as oncolytic viruses or cell therapy. Next to very simple things, such as arsenic or radium, it is remarkable how far back in time some of the approaches go. The first documented attempts at cancer treatment with arsenic date back over 200 years. This very early, very old publication, from 1801, by the way, contains a remarkable statement. It basically says, "The patient died, but the medicine was great nevertheless." Doesn't this sound strangely familiar? Even today, we still get submissions in which the response rate shows a pharmacological effects, but then overall survival data fail to show a benefit. The radiotherapy of tumors also has deep roots. The first publications on the radiotherapy of tumors are almost 100 years old. And here, too, we find that perhaps not so much has changed since then, just as today many innovative drugs are first introduced in the USA. It seems that the particular innovative drive on the other side of the Atlantic was already recognized around 100 years ago. We may also see or suspect a new group or category here, householding metabolism. Admittedly, we still have only a handful of anti-cancer products that use householding metabolism as their primary pharmacological target. And admittedly, this potential group is much more heterogeneous than, for example, the protein kinase group. So, still too early to say for sure whether the targeting of householding metabolism is a sustainable and important trend. Even if the first product was authorized 29 years ago. The first product in this group was the proteasome inhibitor bortezomib, Velcade, at least in the centralized approval. Asparaginase was of course approved decently even earlier. Proteasome inhibitors are another good example of how drugs develop within a class. After the first-in-class lag phase, in this case over 10 years, improved products occur. Velcade is administered parenterally and reversibly binds to its target. Kyprolis, more potent, comes with irreversible binding, and Ninlaro offers the advantage of an oral treatment, while on the other hand it binds reversibly and may seem less potent than Kyprolis. And with that, we are at the end. We will update this overview when new products are approved and will make the updates available. As a bonus, in the last slide, You have a, let's say, enriched version of the overview figure we have used for this presentation.
Thank you for watching and listening, and on the now really very last slide, after the list of abbreviations, you find how you can contact us.
Thank you.
And goodbye.